Okay, so those two assignments, one today at 10, the one on Friday at 9. I'm saying it again because I'm recording now, so that people who are listening, they can go into Educat and make sure to do those assignments. So we're covering the light reactions and trying to get into this idea of how do the plants use that light energy coming from the sun to actually get a chemical equation up and running so that they can end up with products that they want to use for the next phase of photosynthesis. So when you look at the design of most leaves, they tend to have a pretty good surface area. And when you look at a plant through the day, most plants will try to get their leaves in position so that their surface is about 90 degrees to the incoming light from the sun. So that means when the sun rises, a lot of plants will face their leaves to the east and try to have their leaf blade so that when the sun is coming up over the horizon, the leaf blade is getting full on hit with sunlight energy. At high noon, you would see a lot of leaves are horizontal to the ground to get that the sun directly above them, hitting at 90 degrees again. And so a lot of plants will do that. They can move their leaf in order to maximize the amount of light striking their leaf blade. And then if you look inside the leaf, it's really remarkably well designed to allow for sunlight to go through the whole tissue. Like it doesn't just stop at the first layer of parenchyma cells inside the leaf. We see that they actually call it a channeling or a tunneling function of the design inside the leaf so that the light is getting channeled or tunneled through into the deeper layers of the leaf. And it is something that is really efficient getting that light to come through. So most plants, when you look in the leaf, they have those palisade cells that are kind of stacked right next to each other like columns, and they're kind of tightly spaced next to each other. But the light tends to funnel through those. I mean, some of the light's being used by those cells, but then some of it goes through to the spongy mesophyll below. And then remember now, the spongy mesophyll area of a leaf has a lot of air gaps. And what they find is those air gaps allow for bouncing of light all over the place inside the leaf. So you get this tunneling and then this refraction and reflection in the air spaces there that allow for the light to bounce to all the spongy mesophyll cells. So it's actually amazingly well designed to allow for light to come in and really get used through the whole leaf. No one section of the leaf tends to hog the light and use it for its own. The rest is left undry. The other thing too is that leaf tissue is highly vascularized with veins, and you have major veins, the minor veins, the capillary veins, and so you have all of that connecting into it so that the mesophyll cells inside the leaf, that they're not very far away from any one vein. It's very easy for them to make their sugars and dump it into the phloem really easily. And so it's a very efficient way for the plant to do photosynthesis, get the sugar, get that transported. Lots of chloroplasts per mesophyll or parenchyma cell inside. Anywhere from 20 to 60 is a decent range for where that covers most species that they've looked at. So again, having lots of chloroplasts means you can do lots of photosynthesis. Bless you. And then remember that guard cells, depending on the plant species, some guard cells have plenty of chloroplasts, others do not. So that was kind of a surprise. We didn't realize they just assumed all guard cells had chloroplasts. They don't. How many chloroplasts per cell? Yep. And so they, some species can have up to 60 chloroplasts in one cell. So that's a lot. Okay, so really, really well designed. We're going to zoom into the chloroplast. So you've had this information. I'm just reminding you. There's two outer membranes. That outer membrane, the inner membrane. The inner membrane is more selective, so it's selectively permeable. The outer membrane is more sieve-like, and pretty much anything can get through there. within reason. And then remember, inside the chloroplast, there is that thylakoid membrane that's floating around inside. And that's the one that has the relatively stacked regions and unstacked regions. Okay, and so if you crack open a chloroplast and look inside, or the thylakoid and look inside the thylakoid membrane, you call that internal part the lumen, just like in our intestines. The inner part of our intestines is called the lumen. And then you have the thylakoid membrane that's stacked. We call it the grana region. The stroma lamellae is the relatively unstacked region. And it's the thylakoid that we're zooming into now. It's the thylakoid that has the chlorophyll pigments. Chlorophyll A and B are what we're going to zoom in on for most terrestrial plants or commercial crop production plants. We're thinking about chlorophyll A and B. So this is where we have the site of the light reactions is the thylakoid membrane. Now, the stroma portion is the watery portion that the thylakoid is floating around in. And we're going to find that that's the portion that has the job of getting the carbon dioxide coming in and making sugar from that. Unfortunately, this room is going to a lot of bugs. So the stroma, so this is, you've had all of this. This is just a refresher. And the reason I'm putting this in here is that we're going to start using these terms now and describing where certain things are happening. So you need to refresh your memory on these terms. So that's an electron microscope picture. Here's a drawing of it that's a little bit more idealized, but so the entire internal membrane, it's a little, I've had students get a little confused by this. I didn't like how the artist did this. So right here where they should have like circled the whole internal membrane, like the unstacked regions, the stacked regions, all of that is the thylakoid. All right, not just the stacked regions. The stacked regions are called the grana. So it's a little confusing how the artist labeled this. The relatively unstacked regions are the stroma lamellae, that's correct. But the entire internal membrane that's floating around in the stroma, you would say, oh, look at that thylakoid. Okay. All right, so we're going to zoom into the thylakoid, and we have different pigments that are embedded in the thylakoid that are going to be absorbing light energy. So some of these pigments cluster together into what we call antenna complexes. And it's like the antenna, the rabbit ears on your TV. If you try to catch TV signals that way rather than using like a dish network or whatever. And so those rabbit ears on your TV, that's a good analogy to these pigments that will capture light energy, and their job is to capture the light energy and then get it to where it's needed. And so they're showing here the carotenoids, chlorophyll B, chlorophyll A, those are the antenna pigments. And what they're trying to show here is they got hit with light energy. They have that excited them, essentially. That's what they're trying to show here with the energy gradient is high after they've absorbed light. And then they're trying to show that this light energy gets to where it's needed, which here they're showing this P680 idea. Okay, the P680 is called the reaction center. It's one of the reaction centers. Okay, so when the antenna pigments, they kind of surround the reaction center, they absorb this light energy, and then we often say they funnel it into the reaction center. So that energy gets funneled to the reaction center, and that causes the P680 chlorophyll. There's a chlorophyll in that reaction center, and a very special chlorophyll. That chlorophyll gets excited, which means one of its electrons gets boosted to a higher energy state, to an excited state. So that's what we see. That's what they're trying to depict here. And so the way they do this is kind of funky. When they transfer this energy, it's literally transferring energy in the antenna pigment. They don't transfer electrons. So the term they give to this energy transfer is called inductive resonance. And it's kind of hard to wrap your head around. The best analogy I have for that is if you've ever played around with tuning forks in music class, where the instructor will strike a tuning fork and get it humming, and then they bring another tuning fork to it, and then that one starts to hum even though they're not touching. That's inductive resonance. It's an energy transfer. And that's what we see happening in the antenna pigments in the antenna complexes. So if you want more explanation, you have to talk to a physicist, which I am not, but it's a thing, it's a real thing. And you get this energy transfer, and then you get to the reaction center. That energy gets to the reaction center and it excites an electron. And then once that happens, then you can start to follow a chemical reaction. And we'll work through some of that. Okay, so here, this picture, I like it because they're trying to really show that in the antenna pigments, it's an energy transfer. That's what they're trying to show here. All those light green are the antenna pigments. And you see they're transferring all of that energy to the reaction center. And then it's in the reaction center that you get an electron excited. And if everything goes okay, that chlorophyll electron gets excited, and then that chlorophyll electron gets handed off to a donor molecule right there. So to an acceptor molecule. So you often see this reaction when someone says, this is photosynthesis. I need you to appreciate that this is the big picture chemical equation. It's including both the light reactions and the carbon dioxide fixation to make sugars. So it's in total all of photosynthesis. So carbon dioxide, water, C6H12O6 plus oxygen. If I say C6H12O6, what sugar do you think about? Absolutely, I do too. So I would prefer in this class that you actually learn this second equation here. CO2 plus water goes to CH2O plus oxygen. Because CH2O is the generic way of describing carbohydrates. That's like the basic building block ratio of carbon to hydrogen to oxygen for carbohydrates. Because you'll see glucose is not the sugar that's made during photosynthesis. That's why I kind of want you to forget about the first bullet point idea, even though that's what's out in the, the common literature. But it makes you think about glucose, and it's not glucose that gets made, it's sucrose actually. We'll talk more about that. So in order to get this reaction to occur, you definitely need light energy. You need pigments involved to absorb that light energy. CO2 is what we say reduced. The plant takes in CO2 and reduces it into carbohydrate. Water is one of the substrates, so that is actually going to donate electrons in this process, which is phenomenal because water does not like to give up electrons. So we'll talk more about that. We say that CO2 is the electron acceptor in this equation. And if you take carbon dioxide and you try to reduce it into a carbohydrate, it is what we call a positive delta G. Delta G is referring to free energy. If the free energy value is positive, what that tells you is that this reaction will not happen spontaneously. There has to be an input of energy. And in this case, it's going to be light energy that's going to do that. So that's why if you're walking like yesterday when it was raining all day, how come you couldn't just tilt your head up, stick your tongue out and get sugar? I mean, there's plenty of water in our atmosphere. It was raining all day yesterday. CO2 in the atmosphere. Why couldn't we just get sugar from the sky when it rains or snows? But we can't. Even though there's plenty of CO2 and H2O in the air, they're not going to spontaneously react to give you carbohydrate. And so that CO2 reduction, you need a big push of energy, light energy in this case. So if you take that equation and reverse it, like C6H12O6 plus oxygen, and run it the other direction, that's what you're doing right now. If you had any food lately, you're running glycolysis right now. and you need oxygen for that. And that will happen spontaneously. So the oxidation of glucose, you essentially take that equation and run it in reverse. That's a negative free energy. That means it's just going to happen. Yes. Yeah. No, I can look and see if there is something that's been put out recently, because I did look for this, but it's been a few years now. But let me see if I can find anything more like recently, if they've come up with a better visual or schematic. In the notes section for this slide, I did take an explanation from the book that published this picture, and that explanation may help you too when you read it. So try that, but I can see if there's anything else that can show like exactly what's happening. For me, a visual would work pretty nicely if I can find something.
I guess what would also be the starting period.
It's just a cluster of the pigments that are physically close to each other. I like the tuning fork analogy, like they're so close to each other that energy can kind of jump from one pigment to the next without any physical separation of electrons going anywhere. It's freaky. I mean, no, it's not a chemical reaction. Yeah. 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 No, I know. It's really hard. Every time I talk about this, I've talked about this many years. It's still just kind of go, wow, what the heck? It's a very odd. And like I said, especially maybe a quantum physicist might be able to rattle off a really well described example, but I still struggle with it every time I teach about this, because it's an energy transfer. And so, I mean, vibration is energy, right? And that's essentially what you have going on here with the electromagnetic wavelength and the photons. And yeah. All right, so here, when you look at it from the plant's perspective and you say, why is the plant doing this? Now that big picture equation had oxygen as one of the products. We like that from our perspective. That's one of the great objectives, is that it gives us oxygen to breathe, but that's not why the plant is doing it. They don't care about us in that regard. And so it is to get these two molecules. And so we'll explain how this happens, but basically the whole point is to get NADPH and ATP. And the reason they want that is because it's going to be used for part 2 of photosynthesis, which is the part where carbon dioxide comes in and you get carbohydrate being made. So that's their goal. The oxygen is one of the products. They don't really care about that. It's not really meeting their goal of getting sugars made. Yeah, it worked. Did you see that? I was trying to get that to happen like when I clicked on for the bullet point, but it wouldn't do it. Anyway, all right, so Z scheme. All right, so this is just, if you tilt your head and look at it, it looks like a Z. But basically what they're talking about here is that when you have light energy being absorbed, that causes excitation in the reaction center. And so this P680 is the reaction center that is inside photosystem 2. So we're going to see in plants that there's 2 photosystems, photosystem 2, photosystem 1. They're just named that way because they actually discovered photosystem 1 first, so they called it photosystem 1, and then they figured out photosystem 2. So that one's second. All right, so photosystem 2 has a reaction center called the P680. This is a cluster of chlorophyll and pigments and some proteins all clustered together. And it's one of those chlorophylls that will get that energy from the sun and its electron gets excited. So that's what they're trying to show here by the up arrow. They're saying the chlorophyll electron gets excited, and when it's in that state, it's ready to be passed off. And so here, when they write it as P680 asterisk up top here, what that's saying is that electron, that chlorophyll electron is nicely excited, and now it has the ability to be donated off in that excited state. So when you do that and you start tracking the path of that electron, we say that it's energetically downhill for a bit, all right? And so that's why they show it coming down the energy tower here, where you know, high energy up top, lower energy at the bottom. All right, then you see photosystem 1 is also involved. And you see that one is also getting hit with light energy. In this case, it's favoring far red light. So the 700, by the way, that's referring to what wavelength that it really tends to heavily absorb. So that 700 nanometers, P680 it's 680 nanometers. That's why they give them those numbers. It's referring to the wavelength of light. Okay, so P700 is also getting hit with light energy. And again, when you do that, there's a special chlorophyll in the reaction center. Its electron gets excited and boosted to a higher state. So you write that as P700 asterisk to say, ooh, it's nicely excited. And now it's ready to be donating off that electron, which they're showing here. And then you see that eventually NADP plus is going to get that and become reduced. So when the molecule takes an electron, it becomes reduced to NADPH in this case. So these reaction center chlorophylls, that's where we start to actually see the chemical reactions occurring, where you have electrons being transferred. And if it's been a while since you've thought of electron transfer, so these are redox reactions. Hang on just a second. These are redox reactions that we're talking about. That's supposed to be an R, R-E-D-O-X, which stands for reduction and oxidation. Have you heard of some of the acronyms, like oil rig? Have you heard of that one? Oil rig. That one I learned when I was an undergrad because my older brother worked on the oil rigs down in Texas for a while. So it was easy for me to remember, but oil is oxidation is loss. Oxidation is lost, and then reduction is gain. Okay, reduction is gain. So we're talking about if a molecule loses an electron, that's the oil, then it's oxidation. That molecule has become oxidized. If a molecule gains an electron, then it becomes reduced. That's the rig part. What's the other one? Oil rig. Have you heard Leo the lion says grr? That one? Leo the lion says grr. Leo stands for loss of electrons is oxidation. Grr is gain of electrons is reduction. So whatever way you want to think about it, but basically they go hand in hand. If you have a molecule donating an electron, there's an acceptor there somewhere. And so you have this electron transfer from molecule to molecule to molecule. So that's what we see going on here. They're just abbreviating everything a little bit. A couple of other things to think about on this slide. You see over in photosystem 2, water is getting stripped of electrons. The water is getting oxidized or stripped of electrons. So when light energy comes into photosystem 2, part of the process is not only exciting a chlorophyll electron in the reaction center, it's also creating a situation where water can get stripped of electrons. That is phenomenal. You know how hard it is to get water to give up electrons? It is just not wanting to do that. And so to have, this is the only biological mechanism we know of in order to split water. It's incredibly difficult, and plants have figured it out. So what we see here is that water is getting split and electrons are getting stripped off of water and getting fed into the reaction center. So when you think about it chemically, when a molecule loses an electron, like that P680 chlorophyll, when that loses an electron and donates it off, now it's missing an electron. And so it needs to get an electron back so that it can do this again. And so that's where the water comes into play. So that chlorophyll electron gets excited, the electron gets boosted off to the donor, and then it's like turns to water and says, give me an electron, I need one. I got an empty spot here, I need to fill that with an electron. And so water is what we call the ultimate electron donor in this process. The ultimate electron donor, right? Because it's through the stripping of electrons off water. If we don't have that, maintained, this thing grinds to a halt. So it's the ultimate electron donor. And then if you follow this all the way off to the far right and you see this NADP plus over here, we call that the ultimate electron acceptor. Okay, so water is the ultimate electron donor. NADP plus is the ultimate electron acceptor in this whole process of the light reaction. This is another way to look at what we just looked at in more detail. So they put it on an energy scale again. Anytime you see negative values on this kind of energy scale, that means it's excited and it's gonna be, in this case, we're gonna see the electrons getting donated off quite readily to the acceptor molecules. So in this, you're seeing pretty much the whole detail of the light reactions. So you can see all of these abbreviations are different players in the game of accepting an electron, passing it to the next one, on down the line. So this is an electron transport chain process, and it's all kicked off due to light energy being absorbed by that chlorophyll that gets excited in the reaction centers. All right, so I'm going to go through some of this, not all of it, but there's some key parts that I want you to be familiar with, and other parts we'll just kind of gloss over. Right? So what I like to teach off of is this. It's a little easier visually, I think, to look at. And so I'll be teaching off of this. And so what you see here is the light reaction process. So that membrane that they've drawn, that's the thylakoid membrane. You can see the stroma portion they've labeled. The inside of the thylakoid is the lumen. So you see that there's two photosystems, photosystem 2 and photosystem 1. You also see that there are some other big complexes in this transport chain process. So let me go into that a little bit. So we have photosystem 1, photosystem 2, and we're going to see that they operate in a chain or a series. And what you will learn is that through the light energy absorption, you're stripping electrons off of water, which is pretty low energy. What that means is water hates to give up electrons. Chemically speaking, no. Water does not just readily give up electrons. You have to push that with a lot of energy. And then through this whole process, you end up raising the energy level to create this NADPH, which is, the NADPH is what we call a good or strong reducing agent. It's a reducing agent, or a reducing molecule. So what that means is when you get NADPH made, now that is able to participate in the second half of photosynthesis, where it will get put into a chemical reaction pathway, where it's just like, yeah, I'm here, I can give off electrons and I can help this reaction go. And so it's very happy, chemically speaking, to give off electrons into a reaction pathway. And we'll talk about that later. So these photosystems are really complex in the sense that they're very big complexes. They span the entire width of the thylakoid membrane, and they can They have some proteins in there, some chlorophyll, carotenoid pigments mixed in there. And then surrounding that, you have antenna pigments. So the photosystems are surrounded by antenna pigments, and you have the so-called reaction center. That's where we find the special chlorophyll that will have its electrons get excited and get boosted to a higher energy level and get ready to get passed off. So it's actually in the reaction center that we start to see an actual chemical reaction. What I mean by that is you actually get a transfer of electrons from one molecule to another. All right, so going back to this picture, the bigger complexes that you're looking at in this drawing. So you can see photosystem 1 and photosystem 2. Then there's another one that's called the cytochrome B6F. You can see that also spans the thylakoid membrane. It's a really big complex. And then the last one on here is the ATP synthase. So those four are really big. They span the membrane. They don't move. Okay, they're kind of stuck in place because they're so big. But yet we have to have electrons transferring, between them. So if they're stuck in place, but we still need to get an electron flow going, that means you have to have other players in the game that can help deliver. And so we have also mobile carriers that will get involved in this too. They'll mobilize, they'll move, and get the electrons delivered into the next phase of the chain. So all of these, though, the PS1, PS2, cytochrome B6F, ATP synthase, often they're referred to as integral membrane proteins. I tend to say transmembrane because that's what they're doing. They're spanning the entire width of the membrane. Okay. So in this drawing, you're also seeing some of the mobile smaller molecules like that PQ, PQH2. We'll talk about that, the PC, and some others on there too. So we'll get into some of those. So what we're looking at here, and let's start with photosystem 2. It's very standard that when you describe light reactions, you start with photosystem 2. So we're looking at this end of the figure. So what we need to think about is light energy getting into photosystem 2, and it gets to the reaction center. And then the chlorophyll electron gets excited. So then you would write that as P680 going to P680 asterisk. That's the way to say that, yep, that chlorophyll electron got excited. And then if everything goes okay, that electron will get donated off to pheophytin. And pheophytin, I'll spell it all out. Pheophytin, which you can just abbreviate down to pheo. Pheophytin is actually a chlorophyll molecule that's been slightly modified. And so pheo gets that electron. So then you would write it like this. P680 lost an electron, that chlorophyll lost an electron, so you write it as P680 plus to denote it's missing an electron. The pheophytin gained an electron, so you give it the minus. So when you see it written that way, then they refer to it as a charge separation because now you have a positive and a negative. And this is the step where you can actually point to it and say, yep, this is where we see light energy being converted to chemical energy in the form of a redox reaction. Redox reaction just means you have an electron that got donated from one molecule and accepted by another. So that's what they're showing on this figure, where P680 got boosted to that electron, got boosted to an excited state, and then you see pheo is sitting right there, right by photosystem 2, ready to get that electron. Okay, the other key thing happening here is that you have this. So this oxygen evolving complex. Let's go on to that. So this, although they drew it they had to draw it that way to kind of show what was happening, but please be aware that the oxygen evolving complex is part of photosystem 2. It's like right inside there. Okay, it's part of that complex. And it is incredible because it's in the oxygen evolving complex where you see water getting stripped of electrons. And so they typically describe it this way. If you take 2 water molecules, that way you end up with an O2 molecule. So 2 water goes to oxygen and 4 protons, 4 electrons. Okay, so that reaction allows for the electrons to get stripped off of water. And so what's great about that is that the P680 that's missing an electron, that chlorophyll that got donated off, the chlorophyll's like, yeah, I'm missing an electron. I need an electron from somewhere to be chemically stable. And so that is coming from the water. So the water gets stripped of electrons, that electron goes to that chlorophyll in the reaction center. And now it becomes reduced. And now it's like, okay, now I have my full complement of electrons. Now I can get hit with light energy again. And this whole process can happen again. As water is getting stripped off of electrons, another really important thing that happens is that those protons that get created as one of the products, those get put into or dumped into the lumen portion, which is the interior portion of the thylakoid membrane. That's really important. We'll come back to that. It sets up a nice situation for the plant. Okay, so that oxygen evolving complex is still somewhat of a mystery how it actually works. And like I said, this is the only known system that we know of biologically that can take water and yank electrons off of it. It's quite remarkable. And so it's part of the photosystem 2. And that's why, you see it here. They're trying to show it's all part of photosystem 2. And so water is the ultimate electron donor, and electrons come off of water. They go to that chlorophyll that lost an electron, and then that chlorophyll has all the electrons back that it needs. And then when it gets hit with light energy, it can boost off one of its electrons again. So this happens in like picosecond reaction times. It's amazing how all day long it's just so fast. And what we see happening is once they don't show on this particular drawing that pheophytin is there, but it would be like right here. Imagine pheophytin sitting right there, getting that electron, and then its job is to transfer the electron down the chain to the next molecule in the line. And so that's the PQ that they've drawn in here. So the PQ is the next player. So what we see is pheophytin will donate 2 electrons actually to the plastoquinone. This is a mobile electron carrier. It's small. It's a small molecule. And what it does is it needs 2 electrons to be chemically happy, so to speak. But that's not the whole story. It needs 2 electrons, but then it also needs 2 protons. This is really important because what it does, this plastoquinone, it grabs protons from the stroma. And when it does that, plus the 2 electrons, then we say it gets fully reduced to PQH2. So this point of it's grabbing protons from the stroma, that's another key thing that we need to think about. Okay, that's setting up a nice situation for the plant. I'll explain more. Once that PQH2 is formed, then it breaks free from photosystem 2. It releases off and it starts moving laterally through the thylakoid, and its goal is to find a cytochrome B6F. That's what it has to reach because that's the next player in the game that's going to need to get those electrons. Okay, so this PQH2 moves laterally, hopefully bumps into cytochrome B6F. So that's what you're seeing here. And you can see that the PQH2, so it's moving laterally, and then it donates both of its electrons. So here's the 2 electrons that it's donating off. And you can see those 2 electrons go in different directions. So there's one that goes off this route, and then there's one that goes, kind of stays in the cytochrome B6F. So this, one of the electrons, this goes to the iron sulfur. We'll talk about that later. We call that the linear path of flow, because that's the one that's ultimately going to finally reach NADP+. The other one goes to this cytochrome B. This one, that electron is what we say it's entering into the Q cycle. So within the cytochrome B6F, the Q cycle is this PQ H2 donating the electron to the cytochrome B, the first cytochrome B. Then there, you can see there's another cytochrome B, and then there's a plastoquinone in this complex as well. So all of this that they're drawing up on the top portion, that's the Q cycle. One other thing to note, as this is happening, as the PQH2 is coming from photosystem 2, and then it donates off its 2 electrons, by default, it'll also have to get rid of its 2 protons. And how does it do that? It dumps them into the lumen. Really important concept. The fact that you see these protons showing up in the lumen. That's going to create a really good situation for the plant. I'll explain. Okay, so this is showing kind of the kickoff into the Q cycle. So let me explain a little bit more about this. So with the Q cycle, every time a PQH2 comes to a cytochrome B6F, what you see happening as a result, so this is a second PQH2 that's come through, what we see here is that this plastoquinone in the Q cycle, it's eventually going to get 2 electrons. It needs 2 electrons. Any PQ that we look at, it needs 2 electrons, 2 protons to be chemically stable. So you can see here it's partly PQ, when they write it as PQ/dot like that, that means it's accepted 1 electron, but that's not good enough. PQ I like to think of as a hog. It hogs electrons. It's not happy with just 1, it needs 2. So here you see this PQ has accepted 1 electron, but it's like, okay, I need 1 more. So another PQH2 comes laterally, bumps into cytochrome B6F, and says, okay, I got another one here for you. And so this second electron goes through the Q cycle. That PQ, semi-reduced PQ is like, all right, great, now I got my second electron, but to be chemically stable, I need to grab 2 protons from somewhere. And you can see, in this case, this plastohydroquinone grabbed it from the stroma. So there's protons disappearing from the stroma again. And remember, anytime a PQH2 is moving from photosystem 2, to this into the Q cycle, it's also dumping protons into the lumen. So what you see happening is protons disappearing from the stroma because they're getting reacted with molecules, in this case to become PQH2. All right, so you have a disappearance of protons from the stroma. You have protons getting dumped into the lumen. So when you see that happening, what you are really describing is a proton gradient across the membrane, the thylakoid membrane. You have an imbalance of protons. In this case, more protons are starting to show up in the lumen compared to what you see in the stroma. So that's a proton gradient across the membrane. All right, so each time a PQH2 mobile carrier comes along, it's going to do this. It's going to give 1 electron off to the linear route, 1 electron into the Q cycle. And as long as that PQ, when that PQ gets 2 electrons, it says, all right, I got my 2 electrons, I'm going to grab 2 protons from the stroma. And then that mobile carrier keeps dumping protons into the lumen as it's delivering its electrons. So when you look at it, it really allows for an increase in the number of protons that get pumped or dumped from stroma to the lumen. So if you really look at, like, okay, why bother doing this little Q cycle? What's the point? The point is it's allowing for this gradient, this proton gradient to get established. And that's really important from the plant's perspective. Finding a way to create a proton gradient across a membrane. That's a really common strategy amongst all living organisms. We do it as well. We're doing this in our mitochondria right as we speak. And it's really important because it allows us to do something super good. I'll explain. Some people refer to it as an electrochemical gradient. And that's totally true. It's an electrochemical gradient. I tend to say proton gradient. Depends which way you've been taught previously. Okay, so the Q cycle, even though it looks kind of complicated, really when you look at it, it's a pretty slick strategy to get the development of a proton gradient. Questions? I know I just threw a lot at you here. So I really, really, really encourage you to look this over as soon as you can, because if you don't look this over, it's going to fly out of your head and you're going to have a tougher time. So Friday, I'm going to recap this a little bit, shorter recap of this, and you'll be able to ask questions. So hopefully between now and Friday, look this over so that you can feel more comfortable with this portion of the light reactions. And don't forget to do the homework assignments. They're in EduCat. One's due today, one's due Friday.